Hey, hello guys, welcome, Manakam, Mandanam, Namaskaram. Today we are going to start from biotechnology. Okay, it's basic principles and processes only we are going to study. This lesson is the total basics of entire biotechnology. Okay, well, they have given only surface level. We will see a few points, not a lot of points, just a few points that are above NCERT or little bit in depth. We will be just looking at them. So before entering into this lesson, we will have to have some basic knowledge about one organism, that is going to be this one. This lesson is having a direct contact with molecular basis of biology, which means if you haven't studied molecular basis of biology, then you will have to struggle a bit in this lesson. Okay, so we will try to make that struggle as easy as possible. That means as less as possible. Okay, so this is going to be your E. coli. Okay, so this organism is the place where you will be seeing a lot of places. Okay, E. coli. This is, and yeah, we will be going with technical terms and with book derivation, not derivation, what is that? Book definition, everything will be going. But before that, we will just look at this organism, then we'll be going into the book. Okay, so this organism is known as E. coli. What is E. coli? The full form of E. coli is Escherichia coli, which we have already seen in molecular basis of biology. In the first seminar, I've said little bit, okay, just 1 or 2 points about Escherichia coli, where everywhere, okay, in the total biotechnology, in human welfare and microbes, then in molecular basis of biology, then in biotechnology, then in applications of biotechnology, then also in some other places, I think this E. coli is having a very special place because this is such an important organism. Okay, and now we will just look a few things about it. Okay, so E. coli, which is a type of bacteria. Okay, which is a type of bacteria, and then its family. When you are talking about its family, okay, its family is actually Salmonella. Okay, Salmonella. Okay, oh. Okay, and also this one's close relative, E. coli's close relative is going to be your Salmonella typhimurium. Okay, is your Salmonella. It is also another bacteria which is being very closely related to E. coli because they both are possessing in the same family. Okay, it is not family, it's closely related to Salmonella family. Yeah, it's closely related to this family. Okay, related. Okay, so Salmonella typhimurium. One second, as you all know that I'm very weak in spelling. So typhimurium. Okay, so Salmonella typhimurium. Now you can see that here this is the internal structure. So here it has cytoplasm. Here it has cytoplasm. Then very interesting thing that you have to note is it has two different types of genetic materials. Okay, so genetic materials. It's having two different things of genetic materials. That is linear DNA and then the circular DNA. We will also see the difference between these two. Okay, some differences they both have. Okay, and these two are actually completely independent of each other. We will just look at them in a very detailed way itself. Okay, so just after this next slide we will be watching that. So this is also genetic material, but the thing is it is linear. Okay, and this is also genetic material, but the thing is it is circular. Okay, and you can see that here it is, this total thing is nucleoid. Okay, this thing is nucleoid. Okay, now this thing is actually called as plasmid. The circular DNA is actually called as plasmid. What is your plasmid? Plasmid is your circular DNA which is double stranded. This is also double stranded, this is also double stranded. Okay, these two are double stranded. Now you can see that this is what plasmid, right? So this plasmid is actually having a few a few regions. So this region, like see this plasmid, we will also check about it. Okay, I also have here. Okay, so this plasmid is actually being divided into a few regions. We will actually I have not yet entered into this lesson. We are just seeing some basics that we want to understand this lesson in a good way. So that one is for this one. Okay, so it will be divided into regions, and each and every region has its own uniqueness. Okay, each and every region here will be having its own uniqueness, its own role. Okay, and also like this, it will not be that much uniform. If you want this one structure, then it will be like this. Okay, it will not be like this and then this. This is not the way how plasmid looks. Okay, so the plasmid will be looking like this. Wait, I'm very awful in drawing. I will just give you one time. I think in one of my seminars I already said a little bit about plasmid. Oh god, okay, so like this. Wait. Oh god, I'm very awful in all diagrams. Like this only it is going to be. See, I know this is very awful, this diagram is very awful, but this is how the plasmid is going to be. Okay, and when it replicates, see, we all know that this plasmid will be replicating, right? Okay, when it replicates, do you know how it will replicate? It will be forming a replicating bubble. Wait. Okay, so it will be forming a replicating bubble. That means for an example, this is what one plasmid. So this plasmid will be this plasmid will be divided into two different plasmids. Okay, will be divided into two different plasmids. Okay, like this. Okay, so how will it divide? Okay, what is its role play in division? How will it divide? See, that's actually pretty simple. Over here you can see that in just having like this loop in one place, okay, in just one place, okay, it will be forming a bubble. Okay, it will be forming a bubble, which is actually known as, okay, this bubble is actually replicating bubble. Yeah, this is actually replicating. This is actually known as replicating bubble. So this is what that replicating bubble is. Okay, here we all know replication fork. Okay, which will be like this. Okay, Ogasaki fragments, everything we have seen in molecular basis of biology. Okay, where in one place the newly formed DNA will be in this side, and in another place it will be like this. Okay, isn't it? The direction will be opposite, like that. Okay, here in one side, okay, one side DNA will be formed in this direction, and in another side DNA will be formed in this direction. Okay, just opposite direction, one strand of DNA will be formed, and in this one another strand of DNA will be formed. Then okay, and also in this one will be Okazaki fragments, then another will not be, then this will be ligated, then they will be having a bond between them, then you will be getting two different plasmids like this. Okay, and even if you don't understand this process, not a problem, because they will not ask any questions from here. I don't think this is there in our biotechnology of class 12. If it is there now, then don't worry. In the future lectures I will explain this process still more clearly with enzymes. Okay, with the role play of enzymes. Okay, so this is what your plasmid is, and this plasmid can be n number. That means n number in sense it can be even 100. Okay, this plasmid can, for one E. coli there can be 100 plasmids, or not even one plasmid, or 50 plasmids, or 15 plasmids, or one plasmid, or 3 plasmid. Okay, and that number is completely cannot be guessed. Okay, that number cannot be guessed. Just because this is E. coli, you cannot say that we'll be having fixed 3 plasmids. Okay, and these plasmids are mainly used for protection. You can say for protection. See, as I said that this plasmid will be having its own divisions, its own regions. Okay, see this region, this entire region, this entire region. Still now we have not yet entered into biotechnology. We are just discussing the basics. Okay, so I'm just repeating it. So because I know if you have already read this chapter now, then you will be seeing like, why have I started from middle of the chapter? Like that doubt you might have. Still now we have not yet entered. Okay, then also we are having, where is one more thing? Here. Okay, this is another thing. Okay, so this one and then this one. So ampicillin is there, right? So this ampicillin, actually this is known as, okay, this thing, this region, this entire region. Okay, so this entire region and then this entire region is there, right? So this entire region is known as ampicillin resistance. Okay, resistance. Okay, see, actually this area, okay, it will be also having DNA only. This DNA, okay, or this gene will be coding for ampicillin resistance. What is ampicillin resistance? Ampicillin is a solution, okay, it is a substance which has the potential to harm the E. coli. Okay, so in order to protect itself, this plasmid, okay, plasmid is actually a part of E. coli only. Like for an example, if I'm a human being now, then we have neurons and all those organs, right? Like that, this is also a part. But even without this plasmid, E. coli has the ability to survive. There is no such condition that without plasmids, E. coli will be dead. Okay, actually E. coli is E. coli is more likely to be a protective organ. Okay, a protective organ, not the, I cannot say organ, it is a protective cell organelle. Okay, so this one codes for a substance which helps E. coli to protect itself from ampicillin. That is what ampicillin resistance does. Coming to tetracycline resistance, so this is also a gene only. Okay, this is also DNA, this is also gene only, which actually codes for tetracycline. Okay, for tetracycline resistance, sorry, tetracycline. Okay, sorry, there is no such thing as tetracycline. You know, sometimes your brain lacks. So tetracycline. Okay, sorry, tetracycline. Okay, tetracycline resistance. Okay, so what will be this doing? So this gene will be creating a substance which helps this E. coli to survive from tetracycline. Okay, from tetracycline, this is what protecting it. That means what? That means if this E. coli is having one plasmid, okay, for an example, you just imagine that one E. coli is having one plasmid, and then that plasmid does not have, for an example we are talking, okay, if that plasmid does not have ampicillin resistance gene, it is not there. So then in that sense, if it is in ampicillin, okay, if it is in ampicillin solution, and then here you are having this E. coli, and that E. coli is having its plasmid, and that plasmid is not having ampicillin, then this E. coli will die. Okay, will die because it cannot protect itself from ampicillin solution. Okay, I hope everything is clear till now. We will discuss about OP everything in the future lectures. That means, or future lectures, no need of future lectures. In this lecture, even later on, we'll be discussing. Okay. Now we will see the difference. We will see the difference between plasmid DNA and then linear DNA and then the circular plasmid DNA. Okay, then we'll be entering into biotechnology with all those definitions, everything. Okay, I think I shouldn't have started like this, but I have started like this. It's okay. I should have started it from the use of biotechnology. Where are we using biotechnology in our day-to-day life? That's somewhere where if I start now, then we might all have a very good understanding. Okay, so S dot I know. Now here you are having, plasmid DNA. Okay, then you are having here linear DNA. Now, first of all, what is a plasmid DNA? Plasmid DNA, that we have already seen. So if I'm saying it again, then it is going to be very boring. Okay, so plasmid DNA is actually double-stranded. Then it is also circular. Okay, here it is linear. So in its name, even it is having linear. So I'm not going to write that point. Now this is also going to be double-stranded. Okay, so then after this it is actually circular, and then this is linear, and also this can be also circular. Linear DNA always need not be linear, it can be also circular, but there are some other properties that this plasmid is having. That is the thing that differentiates it from this linear DNA. Okay, it can be also circular. It can be, there is no such thing as it cannot be. It can be circular. Okay, then the second point is that this is having no histones. Okay, no histone proteins. But whereas this is having histone proteins. Okay, this is having histone proteins. Okay, so that's one important point. Then it does not have any necessary gene. Okay, no necessary gene. That means without this plasmid DNA also E. coli can survive, or any organism can survive. But this linear DNA has all the necessary genes. Okay, in our 9th standard we have studied that a nucleus is the head of the cell, or controlling unit of the cell, like that we have studied now, or king of the cell, like that we have studied, I think, where that nucleus is the thing that has all the necessary genes. That is the reason why it is the king or head or something like that. Okay, or if in case that nucleus does not have necessary genes, then the total cell will be destroyed. That means if there is no ruler for a kingdom, then how will the kingdom be sophisticated and systematic? It will not be, right? Okay, so this is your third point. Then coming to your next point, it is independent. It is independent of, okay, so it is independent of main genome. Okay, it is independent of main genome. That is what also this point is saying, main genome. But whereas this one replicates along with main genome, because linear DNA is the thing that has main genome in it. Okay, replicates along with main genome. Okay, then here it has no introns. But whereas this is having both. Okay, has both introns and exons. Okay, exons are the thing that is responsible for the expression of a character. Introns is where most of the part of the DNA is actually filled with introns only, like 98% to 99%. Only a small portion of DNA is having exons, which is responsible for the exertion of characters. That's it. Okay, accession of characters. Now we will come to biotechnology, I know, but everything is basic. Whatever we have seen here is basic. It will not come under biotechnology itself. It's like basic biology. Now we will come to, or it's like studying about E. coli cell organelles. Now we will come to biotechnology. What is biotechnology actually? See, biotechnology is an advanced science, we can say it, okay, or no, it's not advanced science, it is basic science only. Basic science where we have integrated natural cells, we have integrated cell organelles and then science together. Okay, you know, the technology, the integration of technology and then cells or cell organelles, okay, is what actually known as biotechnology. Why are we integrating? Actually, what is the need of biotechnology? With biotechnology, you can create viruses. Yeah, we can create another pandemic, we can create another lockdown, then we can again have leave. We can create a new corona. New corona in sense, we can create a new virus by using biotechnology. Then this is actually used in medical field. You know, I will just give you some examples. CAR T cell therapy. Okay, this is actually CAR T cell therapy. This is not in our portion. This is not at all in our portion. I don't even think that's even in books. Okay, but I read about it because I know a person. Okay, see, CAR T cell therapy. What is CAR T cell therapy? This is a very advanced model. Lately only they have introduced this. Recently only in Tamil Nadu, Velur district, in that CMC hospital they have taken this and then they are researching it on people with their acceptance, of course. Okay, but this is actually popular in China, then in some other countries also, but cheapest and in a very good place where you can have this therapy. You cannot have it. Cancer patients can have this therapy is actually China, and it is a very costly therapy also. Okay, because this is very latest model, so that is why. And also in our India, Bombay, they have. Then also in some other places they are having this. Okay, so we will just check a little bit about it. Okay, so what is this CAR T cell therapy? This is actually a therapy that is given to cancer patients. Okay, given to cancer patients. Okay, cancer patients. One second, I will. Okay. Okay. So this is given to cancer patients only. Okay. You know, cancer patients in sense. Wait a second. This is not good. Okay, cancer patients. That too, those who are having liquid cancer, which means blood cancer. Okay, very horrible type of cancer. Cancer is actually of a few types. Okay. Amongst that, blood cancer is the most horrible type of cancer. Okay, so this is actually for blood cancer patients. You know what will they be doing? It's actually genetic engineers. Okay, biotechnology is branch, one of the most branch is genetic engineering, which we will be seeing. Okay, so what they will be doing is they are also like those who play with the genes of T cells. What are T cells? Actually, they will be taking T cells itself, T lymphocytes of the affected patient. Okay, there will be the patient who is actually suffering from blood cancer. From them, they will be taking the T lymphocytes. From thymocytes, they can get it. Okay, because that's the place where T lymphocytes mature. B lymphocytes, in sense, then you can take it from the bone marrow itself. But since it is T lymphocytes, they will be taking it from there. Then they will be modifying the T lymphocyte in such a way where that T lymphocyte itself can attack the cancer cell and it can destroy that. So that's what the latest model. Then again they will be taking the T lymphocyte. Then they will be giving intravenous injection. Okay, by intravenous method they will be injecting it back into the cancer patient's body. Okay, where that will be going and replicating in their cell. Okay, by bone marrow. Then that T cell. Okay, that CAR T cell would have got developed a lot, and then it will be fighting against all the cancer cells. Then that patient will be cured from cancer. And the average time that a patient can take in order to get cure from cancer by using this therapy can be about 3 months. Okay, 3 months. And also they will be watching them in a very keen way. Then after that only they will be deciding whether shall we let go of this patient or should we again torture that patient or not. All cancer therapies are very painful and very brutal. It has lots of side effects, but this is a therapy which does not have that much side effect. And also your immune system will be increased a lot because of this therapy, because the T cells modification, because of the T cells modification. Okay, so I'll not write anything about this. We will just stop this because it's not there. Okay, so coming to biotechnology, we will start from its definition, of course. According to European Federation of Biology. European Federation of Biology. Okay, this is a federation or this is an organization, this is a community, you can take whatever you want. Okay, where each and everything, see, chemistry is having its IUPAC, like that biotechnology is having a thing in Europe, that is actually European Federation of Biology, which has given a standardized definition for biotechnology. That is going to be, biotechnology is the integration, is the integral of natural science See, in CAR T cell therapy, what did they do? They have taken the T cell, which is the cell itself, the whole cell they have taken, and then they have used natural science. Okay, then they have modified that cell by adding some other genetic material. Then again they have given it to the patient itself. So where you can see that they have used natural science. Okay, so they have used the natural science and cell organelles. Okay, cell organelles. Or also they have used also cells. Okay, the total cell even they can use. Okay, like in this case they have used the total cell only, or in some other cases they will be just using a part of the cell, or even in some other cases they will be just using the DNA of the cell. Okay, just the molecule of the cell also they can take. Okay, so that is what will be saying as through of. Okay, so that is what thereof. Okay, so parts thereof. Okay, so parts thereof means cell organelles like plasmids only. Then molecular analogs. Okay, then also molecular analogs like molecular structures like DNA and RNA, like that you can take. Okay, well, we need not to write this also again. Okay, so okay, so now next also there will be molecular structure. There will be molecular structure. Okay, molecular structures also. So where they have combined natural science and then and here and is very important. Okay, and any of this part. Okay, any of this part. So they have combined natural science and any of this part. That is what it will be saying as European Federation of Biotechnology. Okay. Now this biotechnology's definition is actually quite simple, isn't it? It is quite simple. Okay, so now we will be just going. Actually, yesterday night I was filming a physics video, and somehow, for the first time, I felt like I wasn't filming very good. Actually, theoretical part I'm okay with physics, but in the numerical part I'm very weak. Okay, so and also any seminar you can take any formula, every formula is theoretical part only. So that was the first time where I felt like, oh God, I cannot even explain it properly. Okay, so coming back to this, I don't know why am I saying that. So biotechnology. See, biotechnology is having two main important streams. So biotechnology is built with these two important streams only, and my personal favorite thing is genetic engineering. Okay, so two different streams are going to be genetic engineering. Okay, it's going to be genetic engineering. Genetic. Okay, so engineering. See, actually, this is personal data only, but it's okay. In my college time, I'm going to take this only. Okay, so genetic engineering, I'm going to do PG in this only. Okay, but in doctor field. Okay, not human doctor, another doctor. Okay, so genetic engineering, and then bioprocess engineering. Bioprocess engineering. Okay, engineering. You know, engineers. Okay, one of the most pitiful souls of this present timeline. Okay, we will come back to your event. So biotechnology, where biotechnology is actually of genetic engineering and then bioprocess engineering. Now come into genetic engineering. Here you are having, what is genetic engineering actually? Genetic engineering is the technique. Okay, is the technique. Is the techniques to alter the chemistry of genetic material Okay, genetic material in sense what? Genetic material in sense DNA and RNA. Okay, so genetic material of DNA and RNA to introduce these into host organism. Okay, to introduce these into host organisms. Okay, so this is your genetic engineering. Okay, coming to bioprocess engineering. What is bioprocess engineering? Here actually you are modifying the genetic material, but whereas over here you are not modifying the genetic material, but instead of that you are actually cleaning the surface. Okay, Kooti thodachi periki badarma vekkiradu. That's it. So you are having, see, any research wants a place to do research, isn't it? So that particular equipment's cleaning thing you can say as bioprocess engineering, and widely it is said as, okay, just a basic term. Okay, a basic term that is used in many places is sterilization. Okay, in hospitals and all, they sterilize each and every object before doing operations and specialized missions. Okay, so that is what, and if in case they don't sterilize it properly, then you can do medical negligence. Medical negligence, no? Medical negligence only. It is there in medical book college. I've seen it once. Because there is a person whom I know studies doctor, but I don't know why am I saying all these things. Somehow I will be mocked. Okay, so bioprocess engineering. What is this? This is the maintenance of sterile ambience. What is ambience? Generally, this is a very good term that we all are using in our day-to-day life. That is, for hotels at all, if you are going now, then if you are seeing the hotel's structure, like the wall color, then the table model, then also the lightings, all those effects inside the hotels that we have, right? That we will be calling together as ambience. So that is what it here was. Ambience, surroundings, okay, all these we can say it as ambience, but the thing is, it should be closed in all the sites. That's what we'll be saying it as ambience. It should be closed, or it should have the ability to be closed, okay, in all the sites. That is what we'll be calling it as ambience in chemical engineering process. In chemical engineering, okay, in chemical engineering process, so that no, we will just go along with book definition itself. Okay, to enable process to enable the growth of only the desired microbe or eukaryotic cell, okay, or eukaryotic cell in large quantities, okay. See, actually bioprocesses engineering is done in microbes, okay, in human welfare. I have said this, that is, if you are having here, okay, an object, okay, here you are having, okay, here is the place where the vessel is being placed, okay, and here you are having the agarose gel, okay. In that agarose gel there is bacteria. Now if a fungus is growing, now then this bacteria will be, okay, this bacteria will die because of the fungus, okay. So like that, here you cannot let these organisms to be affected. The organism that you are trying to grow, actually when they will try to, this is actually a vessel, okay, the cleaning of vessel you can say, okay, in normal English you can say it as cleaning of vessel. After doing all the processes in genetic engineering, they will be going to bioprocess engineering, so that they can reproduce that particular organism, and then we can have many organisms just like that, okay. See, here this is an cell, okay, this is a cell. I don't know why my English is very awful today. This is a cell. Now this cell is actually, you can see that they have done all those operations, they have done all those genetic engineering operations that is required to be done, so that is done. Now what they are doing is, actually they are trying to replicate, okay. To replicate, it has to be kept in a medium, okay, in agarose gel. Normally they will be keeping it so that it can replicate, okay, and then you can see that 3 different cells are being produced. Okay, so to produce multiple copies, they are using bioprocess engineering, and they cannot just they cannot just give up on the cell. That means, for example, if the cell is dying, then there will be a huge loss for the company, because they would have spent lots and lots of lakhs, we can say, okay, they would have spent lots and lots of lakhs to alter the cell's nature in a different type. Okay, to alter the cell's nature even, they would have spent a lot of money. So that's the reason why they have to be very careful, and that's one of the most important reason why there is a separate engineering called as bioprocess engineering. Because generally this we would not study in a separate way, but just because to save the cost, we are having a separate engineering called as bioprocess engineering. And believe me, comparatively, this is easier than this one. Okay, because here only you will have to use all those important things, but here it is sterilization, keeping it all good. But here also there are some complicated things, but we don't have any of those. Okay, so this one we have seen. Now what we are going to see is, so now we will go into book context. Okay, so for how much time am I recording this video? What's the timeline? Okay, so 41 minutes, still I have 20 minutes. So which means we can complete it till 9.2. Okay, so we will complete it as much as possible. I don't know how much we can complete. Okay. So we all know that reproduction is actually of two different types. Reproduction. Again, still we are in the applications only. We are still in the applications only. This biotechnology is actually done in our day-to-day life also. See, even mothers, all the mothers, okay, all those who are in cooking are actually a person who are involved in this technology regularly. Making of curd, making of bread, making garlic naan, making, what is that known as? I've made that many times. In Domino's they will be selling it. Okay, so I've forgotten the name. Great. Okay, so making all those things is actually a part of biotechnology only. Anything that you alter, okay, if you are altering gene or if you are altering a cell by using technology, then you can say it as biotechnology. Yeah, and also you can modify the gene. In day-to-day life, they are using in a lot of places. For an example, also recently they have taken the gene of jellyfish, which is responsible for its glowiness. Okay, the jellyfish is glowing, right? For that, a gene is what responsible. They have taken that gene and then they have induced it into a plant cell's gene. Okay, and then that plant was actually glowing in a few parts during night time. That's a very special thing. Like that you can say. And also, if a baby is having, sorry, if a zygote is being detected with some sort of disease in the earlier stage itself, then these genetic engineers can very easily rectify that. Okay, so for all of those, even for rectification of diseases also they are using genetic engineering. Okay, so now reproduction. See, reproduction is actually of two different types. You might think, why am I saying about reproduction? We will just check about it. Okay, sexual reproduction. Okay, so sexual reproduction and then asexual reproduction. Asexual reproduction. What is sexual reproduction and what is asexual reproduction? This we all know, right? So why am I explaining this one? This is a place where okay, in sexual reproduction is the place where variation will be happening. Okay, variation will be taking place. Variation of characters, you can see. Okay, so that will be taking place, and this is a place where genes will be stored. Okay, you can say that no variation will be taking place. Here it will be all saved. Okay, there is also a term that they have used. In asexual, preserves. Yeah, preserves the gene. Yeah, preserves the gene. Very basic English. Preserves the gene. Preserves the gene, but here it does not preserve the gene. Instead of that, it gives a variation to us. Okay, so also we can say that hybridization. Okay, hybridization, which we have studied in sexual reproduction of plants, and also in animals. Hybridization is done. For an example, hybrid dogs. Okay, so Labrador is created in the lab. Like that, there are hybridizations is done. So in that time, what will be happening is you cannot expect. Okay, you cannot just expect to have only good qualities. Okay, both good qualities and bad qualities will be there. Okay, so both good quality. It will have also good genes. Okay, good genes which are known as desirable genes. Desirable means you want something. If you want something, you can say that I'm desiring for it. So that's what desire mean. Okay. So next is a bad gene. Okay, or negative impacting gene. You can say. Okay. So or unwanted gene we can say. Okay. Bad genes. Okay, genes which are actually unwanted genes. Unwanted genes. Now you will have to separate this bad gene from this good gene. For that, what you will be doing is to separate this bad gene from this good gene. What you will be using is you will be using biotechnology. Okay, so you will be using biotechnology in sense recombinant DNA. In that time, you will be changing this DNA to have only good gene. Okay, so that DNA is known as the changed DNA. You will just change the DNA. Okay, change in DNA. There will be a change in DNA. So that change in DNA is what we are saying as rDNA. Okay, recombinant DNA. So recombinant DNA. So recombinant DNA. Why has it got its name as recombinant DNA? It is very simple. That is, you are having a cell, okay, and the cell is having its own genetic material. Okay, so now one part of the cell, okay, in this genetic material, one part of this genetic material will be cut. Okay, so then it will be like this. Now in that time, you will be inserting another DNA which will be combining. Okay, recombination. Okay, again it recombines with here. Okay, this time it combines with a thick thing. Okay, so that is why we will be saying it as, since it is being recombined, so we will be calling it as recombinant DNA. Okay, so there is recombinant DNA. This is what actually we will be calling it as recombinant DNA. And also for this they will be using also, along with this they will be also using DNA cloning. Okay, DNA cloning is done, and DNA cloning is done by PCR method. Okay, by PCR, which is having, Thermus aquaticus. We will also see about it. If in case it is there in our portion, then we will be seeing it in a very detailed way also. Okay, so Thermus aquaticus is there. So Thermus aquaticus is a bacteria that is archibacteria, so that survives in lots of harsh conditions. It will be surviving even in the hot streams of volcano. Okay, not in volcano, they will be nearer to volcanoes. There are some lakes, so due to the heat that is coming from volcanoes, those lakes, okay, that will not be in that much big size, they will be in small size. Okay, so those volcanoes are known as hot springs, and they will be very hot. That hot spring is actually in those places this will be available, and this will be producing an enzyme called as Taq polymerase. Okay, about this we will be seeing later. So that Taq polymerase is something where we will be using to clone DNA. Okay, so DNA cloning is a place where it is being used. Then after that, gene transfer, also in gene transfer. Okay, also in gene transfer we are using it. Okay, so gene transfer, we are using it. Oh, over here we will not do anything, just we are talking about over here. Okay, so this is a place where, this is also one place of application of biotechnology. Okay, so now next, after this, what we will be doing is we will be seeing about the fate of DNA. Okay, so what is the fate of DNA? What is the sentence fate of DNA mean? See, the thing is very simple. This is your plasmid. Okay, this is your plasmid. Okay, so you want a specific gene. You want a specific gene to be replicated. See, what are genes actually? First of all, what are genes? Okay, what are genes? What are genes? What are genes? Genes are responsible for projection of characters, for expression of characters. Anything that happens in your body, genes are what responsible. By the process called as central dogma, Okay. So by the process called as central dogma, what will be happening is DNA, okay, it will be going first replication, okay, it will be going first replication, then transcription. By transcription process, it will be formed into RNA. Okay, so RNA by the process called as translation, proteins will be formed. What are proteins? Proteins are those which are responsible for the production of hormones. Hormones. Okay, so hormones. Sorry. What am I writing? Enzymes. Okay, so enzymes are responsible for your characters. Responsible for your characters. That means if your characters, then also some biological processes that are happening in your body. Okay, biological processes that are happening in your body, and also insulin is and also some hormones, and also all the hormones are produced. Okay, then also hormones. Okay, hormones are being produced by genes. Okay, genes have the knowledge. They are like the book. Okay, they have all the knowledge. By using that knowledge, some cells, okay, so some cells use that knowledge, and by using that knowledge they produce the product. Okay, that is hormone. For an example, now day-to-day life, insulin's demand is being increased a lot. Okay, so like that also you can take. So for that you have to take insulin's gene. Okay, insulin's gene. So insulin creating gene you have to take, and that gene you have to replicate in a cell, then automatically that cell will start to produce insulin. Then that insulin you can use for yourself also. There is no problem with that. But do not use it if you don't have any problem. Why to fix something if it is not broken? It's a very common say in English, right? Okay, so now we will come back to here even. DNA cloning, like we can literally summarize entire biotechnology within this one slide itself. Okay, very beautiful slide this is. You are having here, okay, a chromosome. This is an eukaryotic chromosome. What is eukaryotic chromosome? A chromosome that has been taken from an eukaryotic cell. That is what we'll be saying it as eukaryotic chromosome. So this chromosome will have your desired gene. So chromosomes are what? They will be having that desired gene, right? So this chromosome is the thing that is going to have that desired gene. So that desired gene alone you are going to extract it. Okay, see, DNA fragments of interest. Okay, DNA fragments of interest. That means the desirable gene. That is what it will be saying as DNA fragments of interest or DNA of interest. Okay, is obtained by cleavaging. Okay, cleavaging is a process by which you have obtained. Okay, you have obtained this DNA fragment from this chromosome. Okay, then chromosome with restriction endonuclease. See, this restriction endonucleases is very important. Okay, we will be talking about it in our upcoming time. Okay, now I will be not giving that much. I will just say that this is a substance. Okay, this is a nuclease. This is a substance, or this is a nuclease which is used to cut DNA. Okay, this is used to cut a piece of DNA. That's alone enough for now. In later on, okay, in the future we'll be talking a lot about this. Okay, this restriction endonuclease is actually, see, first of all there is nuclease. Any enzyme or any substance that cuts this, okay, any enzyme only, it has ACE now. So any enzyme that cuts DNA or that cuts genetic material will be called as nuclease. Okay, so amongst that there is a specific categorization, that is restriction enzyme. So this restriction enzyme is actually which will be produced by the cell itself. We will be also talking about it, why the cell is producing restriction enzymes. There is no problem with that. Okay, so restriction enzyme, and this restriction enzyme is actually of two different types. That is restriction endonuclease, okay, and then restriction exonuclease. So endonuclease, exonuclease, both are used for cutting only. This is also known as molecular scissors. Okay, this is known as molecular nibblers. What are nibblers? I have one, I think. Nibblers are like this. Okay, they just cut from the end. You cannot just like that, cut it from the center. You will have to cut it from the end. Okay, you will have to cut it from the end. So that is what we will be calling as nibbles. Okay, so molecular nibbles. These are known as molecular nibbles. What are best nibbles? I had one in my home event. The best nibbler I ever had is my rat, and now my bird is trying to gain its place. It had recently passed away, so now it's trying to gain its place. My bird is also biting in a very good way. It's also nibbling in a very good way. Okay, so this one, okay, by using this, mostly we'll use this only. Okay, we will use mostly this only, but we will not use this one that much. So this endonuclease, and also that will be giving you sticky end, blunt end. That will be all seeing in the restriction enzymes in a very clear way. We will, for now, you just keep like it. This will be cutting. Okay, this will be cutting the DNA. Okay. Now this will cut the DNA, and that DNA, see, this is the DNA that is being cut. Okay, so that DNA will be inserted into the plasmid. Okay, inserted into the plasmid. So restriction endonuclease is actually of many different types. Just like in dog, how many categories you are having? You are having pug, you are having Doberman, you are having Rajapalin, you are having Chipipara, you are having like that in many categories, right? Like that, restriction endonuclease is also having many different categories. So that same, okay, if for an example, let us take that you have used a particular restriction endonuclease. Okay, for an example, you can take EcoR1 or you can take HIND2, anything. Okay, we'll take HIND2 even. Okay, so or EcoR1 shall we take? Okay, you take any one restriction in, okay, any one restriction endonuclease, that endonuclease only you would have used here to take this particular gene. That same endonuclease only you have to use on this plasmid also, and when you use on this plasmid, automatically you can see here there will be a break. Okay, you can zoom and you can see here there is a break. Maybe you cannot see it that much clearly, but there is a break. Okay, there is a break, and here exactly in this place you can insert this, okay, by using DNA ligases. I'm just giving you all one liner. You will see about it in a very clear cut way itself. Okay, DNA ligases. Okay, DNA ligases is there. This, now what you are going to do is you are going to insert it into cell. Okay, and that insertion is done by, okay, some methods that also you will be seeing. Okay, now you will be inserting it into a cell. Now this cell will actually have to, every organism in this world will try to replicate. Okay, so in order to maintain the count of its species, okay, that cell will be replicating itself. You know, even for humans, humans have that sixth sense. So because of that, according to them, their generation, their bloodline, okay, what can we say? So their feelings should be living. So for them, their live feelings is actually their generations, their offspring. So that is why humans reproduce, not in order to save the number of species. Already there are n number, but there are also some other organisms which are replicating, sorry, which are reproducing to maintain their species on this planet Earth. And it's like the God had created them to have that urge and to reproduce. Okay, so that only the ecosystem, everything will be sustained. If in case a species is being destroyed, then in the food web there will be another species to replace that, but in the food cycle there will be a problem. Okay, we will come back to here even. So here you can see that. Okay, here you can see that. Now this is the cell. Now this cell will be replicating, and while replicating without its knowledge even, what it will be doing is, it will be replicating along with the DNA of this, okay, this desirable chromosome. And this is done by lots of scientists, by genetic engineers, but always nature is the best. Do you know which is the best genetic engineer? That is our bacteriophage. That is our bacteriophage. Such a criminal organism this is. Okay, bacteriophage is a very criminal, okay, very cunning fellow. That bacteriophage will be there, na? So bacteriophage will be looking like this. Okay, So this will be having its own DNA. This DNA will insert over here, then it will go here, then it will be combined over here. Then this cell will be replicating the DNA of the bacteriophage. Okay, and then this DNA, okay, for an example, let us take, okay, for bacteriophage, and then this DNA that we have studied in our 11th even in the biological classification. Now we will not discuss about it. Okay, so for an example, you just take this bacteriophage. Okay, this bacteriophage's case we will close. Now we will come back to here. And for an example, you have given insulin's gene. So if you have given insulin's gene, then in that time these cells will start to produce insulin. Okay. Okay, I know my handwriting is very horrible, awful. Okay, they will start to produce insulin. So this is what your basic one-liner of total biotechnology even. Okay, we will come back to here even. Okay, I am trying to complete at least till 9.2. I thought I will complete everything, okay, till at least half of the chapter. But I am explaining, talking because, you know, today I am kind of energetic. So because of that. Okay. So now we will come to fate of DNA. Real fate. Fate of DNA. What is the fate of DNA? What is that sentence? What does that phrase mean? That phrase means that what will happen to the DNA if you just insert it into a cell. Okay, by the way, insertion also we will be studying. Okay, component DNA we will be studying everything. Now we will come back to here even. It's actually very simple. For an example, see, first of all, we will just check why to insert a DNA. Okay, why to insert a DNA? Okay. Why to insert a desirable gene? See, we are actually inserting desirable gene so that we can get, see, actually no gene, this is the answer, no gene can replicate, okay, can replicate on its own, okay, because, why so, why, okay, why no gene can replicate on its own? Because, see, for replication of gene, there are a few specific typical content that is required. By using those resources only a gene can replicate, and that resources will be produced in a cell only. This gene cannot produce that resource by itself. Okay, that gene will be produced in a cell only. So that is why we are inserting it into a cell, and that cell and also, okay, and also no gene can replicate, okay, can replicate without ORI. ORI is a part, already I have said in molecular basis of biology. ORI is origin of replication. Okay, origin of replication. ORI. Origin of replication, correct. So ORI only. ORI. Okay, so origin of replication, where this is actually the controlling unit of replication. For an example, I said right. Here we have. So, for an example, this is the, you just take about this is the plasmid. You just think like this plasmid is not there. This is also not there. So this plasmid will be there, right? This plasmid will have ORI in it. This ORI will actually, what it will do is, it will also have nearer to this ORI, there will be ROP. Okay, there will be ROP. So this ROP will create, this ROP will create some proteins, okay, that controls ORI. Okay, so for an example, this ORI starts, okay, this ORI will start replication by DNA, okay, by an enzyme. Okay, it will start, what's the name of the enzyme I told you? DNA transformation, no, DNA replication, that's very important name actually. Okay, so it is actually DNA polymerase. I don't know how did I forget it. It is DNA polymerase. Okay, so this DNA polymerase is there now. So this DNA polymerase will actually help in the replication of this DNA. So what will be happening is this ORI is actually having something called as copy number. This copy number is where the thing that decides how many young plasmids should be produced. So from this plasmid, how many new plasmids can be produced? That is actually controlled by ORI only. So now, since it is having copy number as 2, you can see that it is having 2 different plasmids. Okay, it is having 2 different plasmids. Okay, so that's now this is one thing. Okay, so this is the thing, that is when a host gene, when an alien gene is also known as desirable gene or anything we will say, but combinedly we will be calling it as alien gene only. Okay, because the gene always need not to be desirable. It can be sometimes undesirable also. Okay, so for that you will be actually having your alien gene. Okay, for an example, the bacteriophage induces its gene into E. coli. So in that time, it is not a desirable gene for us. It is a desirable gene for bacteriophage. But we have to say whether it is desirable or whether it is not desirable with respect to us. Okay, so that is why we will be calling it as alien gene in total. So alien gene, whenever it is being put into host gene, okay, into host gene. Okay, so host gene that is actually having multiple, okay, that we will be normally taking, which is having multiple ORI. Multiple ORI. Okay, multiple ORI. Sorry, multiple copy number. Any plasmid will have only one ORI. Okay, that. Okay, now you have to forgive me. So that will be having ORI. So where this ORI will be initiating the replication that I already said, but I'll just write it. Okay, initiation of replication happens. Initiation of replication happens. So this ORI will have something called as copy number. So that copy number is equal to number of plasmids produced. So always we will be having the need to have lot, right? The greediness never leaves us. So that is why we will be always taking an organism which has high copy number. Okay, so that lots of plasmid can be produced. So lot of output we can get. That means lot of insulin or some other desirable product we can get. Okay, lot and lot. So and also this one. Very important thing. This is actually an alien DNA. Okay, so wait a second. An alien DNA is linked. With the origin of replication. Is linked with the origin of replication, so that this alien piece of DNA, can replicate, okay, and multiply itself, itself. Okay, and this is what known as cloning. Okay, and then this is what known as cloning. Okay, so this is actually very simple. That is any alien DNA that is being inserted into a plasmid should be first going and connecting into a plasmid. Then with the help of ORI only it will be able to reproduce. That means it will be able to replicate, and that process is what known as cloning. Actually, replication or reproduction of any organism, okay, without any change of its parent is said as cloning. Okay. Now let us take for the first instant of artificial recombinant. Okay, creation of first artificial recombinant. First rDNA creation. First rDNA creation. Okay, so it is done by two different persons in the year of 1972.